Students, welcome to Short Upstairs classes. So in this lecture we will be starting with the IITs and Biotechnology enzymes, and in this lecture we will be here to work of an enzyme that will be the part of the discussion today. For an enzymatic reaction, first a substrate must bind to the enzyme, and there is a binding site that is called active site. Binding site of enzyme is called active site, and to get activated there is a required amount of energy is called activation energy. Active site. Now here in this active site the substrate will get bind for a enzyme substrate to form a enzyme substrate complex. Now here what happens, there are some weak bonds that this substrate made with the active site, and these weak bonds are like hydrophobic bond, hydrogen bond, ionic bond. These are the weak bonds. Ionic bond. Now for these kind of bonds there is required a high energy that these weak bond releases at the time of binding is called as binding energy. That is del G, there is a free energy suffix B for binding. So this is the binding energy, free binding energy that is required to get attached with the active site to a substrate. Okay, now this binding energy is mainly responsible for some increasing reaction rate, increase. Okay, and as it lowers the substrate entropy, and this lowers the substrate entropy, or there is a cause of a conformational change in the enzyme. Conformational change in enzyme. And that facilitates the formation of the ES complex. These facilitates the ES complex formation. Moreover, it also accounts for the specificity of the enzyme for their substrate. Always they should be specify the specificity of the enzyme. There are specified enzyme for the substrate. This is very much important. This is a very much important thing to remember about the enzyme substrate complex or the enzyme substrate formation, complex formation. The specificity is very much important, and that mainly arises from the hydrogen bonding, which is directional and also due to the shape of the active site. It is directional and that is for the shape of the due to the shape of the active site. and that bind to substrate and that have sufficient complementary shape. So this is all about the work of the enzyme. Now after this enzyme substrate complex formation, and from enzyme substrate complex formation the enzyme product complex formation, and from there from that complex the separation of enzyme to the from the product and forming the total product from these enzymatic reaction. Another things that actually contributing to decrease in the activation energy, that is there are some factors, and these factors are really important to know, because the lower activation energy you needed, the higher will be the reaction rate. So there will be more reaction rate, so the more product you will get from a very less amount of substrate complex, substrate enzyme complex. Okay, so let's start with the factors that are affecting to decrease in the activation energy. So the factors that contributing to decrease in the activation energies, there are 4 factors, 4 general mechanism that accounts for the ability to enzyme activity or the catalytic enhancement of a rate of chemical reaction, that are catalysis by proximity, catalysis by proximity. Second is acid base catalysis, acid base catalysis. Third one is catalysis by strain, catalysis by strain. And the fourth factor that is affecting the activation energy is the covalent catalysis, covalent catalysis. These are the 4 factors that helps to decrease in activation energy. Okay, so the let's start with the covalent catalysis. This is the process when covalent catalysis that actually involves in the formation of the transient covalent bond, transient covalent bond. Okay, this transient covalent bond is between the enzyme and the substrate, enzyme and the substrate. The modified enzyme then become a reactant. This modified enzyme that will be then reactant. However, these chemical modification of these enzyme is a transient. That is, this means for a very short period of time, and on completion of a reaction, enzyme returns to the original unmodified states. Enzyme returns to its original state. So this is the transient state. Okay, so again I'm just repeating it, like transient covalent bond is the reaction between the enzyme and the substrate, and this enzyme substrate, in this reaction enzyme will get reactant. Okay, that will modified and will get back to or will change to the reactant part. Now when this is happening, that is for a short time of period, and then after the full procedure the enzyme will return to its original state and there will be no change. That is we call as transition period. Okay, so here the covalent catalysis is particularly a common among the enzyme, and that catalyzes a group that transfer the reaction. So here the covalent catalysis often shows the ping pong mechanism in which the let me draw you this mechanism. Suppose this is the substrate 1 that is reacting with a enzyme, the specific enzyme, and then will form the enzyme substrate complex, ES1 complex. Then from the ES1 complex, EP1 will produce. EP1 means the product 1 will get produced. So from these part, product will get separated and enzyme will get separated from this complex. So it will form P1, and from here again we will get a free enzyme. This enzyme will act on a different substrate, that is S2 suppose. So from these E and S2 we will get a complex, that is ES2 complex. Okay. So from ES2 we will get EP2. We will get EP2. Now from EP2 again, from this complex we will get the product P2, and again the enzyme will get free. Okay. So this is the mechanism that is happening in this state. So this is the transition period. Okay. Now here the tertiary from the this common enzyme mechanism, there is another thing that we can get, another type of substrate molecule binding we can see, and that is like suppose this is enzyme and that is binding with the substrate. So this is S1 and simultaneously it's binding with the another substrate, that is S2. So this is plus and this is also plus sign. Now from these enzyme substrate complex we will get this will look like ES1 S2. This is the complex that will form from this type of mechanism. Okay. So from ES1 S2 again we will get EP1 means E product term from the substrate 1 and the, so from enzyme substrate complex or enzyme substrate mechanism we will get EP1 enzyme product 1 product 2 from the second substrate mechanism. Okay. So from these reaction we will get EP1 P2. So after these procedure we will get ultimately product 1 plus product 2, and from here enzyme will get separated from this entire complex and enzyme will be free again and ready to use for the another biochemical reaction. So this is what happens in the complex, and these type of complex are called ternary complex. Ternary complex. So this was the covalent catalysis. Covalent catalysis when the transition of the catalysis, the catalytic activity, transition of the catalytic activity occurs. Catalysis occurs. So the third category of these work of enzyme, that is catalysis by strain. The third one was catalysis by strain. Now what is catalysis by strain? This is the binding of the substrate to the active site of the enzyme that induces the strain into substrate molecule. That induces strain into the substrate substrate molecule, and this substrate molecule weakening the target bond. This substrate molecule that weakening the target bond and making it more susceptible to cleavage. So this is how catalysis by strain works. Now the third one is done, and the second mechanism for the activation energy, decrease in activation energy, is the acid base catalysis. Now what is acid base catalysis? When acid and base is acting as a catalyst. So let's see what is acid base catalysis. So when enzyme increase the reactivity of a substrate, then this is happening by increasing the electrophilic or the nucleophilic character. And this electrophilic or nucleophilic character, which is actually done by the addition or the removal of the proton, and this proton is actually, this is the mechanism, that is, this proton is donated by, generally provided by acid or base, and when this acid or base is provided this proton to get removed or to get added for a electrophilic or nucleophilic characteristic increase in the enzymatic reaction, then the enzyme activity increases the reactivity of a substrate. So that is why we call it as acid base catalysis. So the first mechanism, that was the catalysis of a by proximity. Now what is the catalysis by proximity? The first one was the proximity. Proximity is the mechanism, this is the idea of the this is the kind of effect, that is the idea to the proximal distance, the distance between the enzyme and the substrate, or we can say that is the enzyme and the substrate when they are actually very much close together in a appropriate orientation, then the bonding is done, and then the enzymatic activity we can see. Enzymatic reaction or activity we can see. So that is why we call it as the catalysis by proximity. So this was all about the factors that are contributing their effect or character to decrease in activation energy for an enzymatic reaction. So thank you for watching this video.